We have quite a bit to get to. We have to start. We're going to go through the structure of cardiac muscle, right, and how it is different than skeletal muscle. We're going to talk about some of the differences in terms of their resting membrane potential, and then we're going to talk about how the heart is electrically stimulated. Okay? So when we talked last week, we talked about the structure of the heart, and we said the heart has a two-sided pump. Okay, something like this, and then the other side, right? And then what is the difference between the left side and the right side? Go ahead.
One gets oxygenated.
One gets oxygenated blood, one gets deoxygenated. Let me rephrase. What's the difference from the left side of the ventricle, left ventricle from the right ventricle in terms of muscle?
Well, the left ventricle has an increased muscle capacity because it has to push the blood to the body.
All right, so it has a increased muscle capacity. What is bigger?
What is bigger?
What is technically bigger? The myocardium, right? Yeah. So the myocardium, remember the myocardium is that, right? And then out here, what's this layer of the heart? That's the epicardium. And then in here, the inside layer is the endocardium. That's an cardium. Endocardium. All right? But we're going to be talking about the myocardium today because that is what is going to do the contraction, right? That's where the heart muscle is, right? The muscle cells, the cardiac muscle cells that are going to be contracting. And so this is what cardiac muscle cells look like. What do we know already about cardiac muscle cells? All right, they are long cells. Yep. Okay, in some cases they can be branched, right? And they have attachments to one another. Kind of look like this. They attach this to each other, right? They're usually uninucleated. What else? What else? What else do they look like? They have intercalated disc, yes. What else? What am I not drawing in here? Striations. Okay, so they have striations. So, kind of looks like that. So, there's your cardiac muscle cell. Okay, branched, uninucleated, striations. Okay, intercalated discs at the boundaries here. Okay, intercalated discs. What's the importance of an intercalated disc? Why are they so special? Emma?
Keeping them together and also
Communication. Okay? These two, so they're going to, what does this heart muscle have to do? These muscle cells have to contract, and all of the muscle cells have to contract at once. Okay? So the intercalated discs are they're a collection of two different cellular junctions, okay? Gap junctions, which allow for these two cells or the multitude of cells to talk to one another in terms of communication of ions are electrically going to move from one to the next, okay? And then also there's desmosomes, which are like the cytoskeleton is actually connected to one another, so that when they contract, they will pull on each other. All right, and we'll talk about that in a little bit. But that's my drawing of the day, my lovely heart muscle cell. It's pretty good, right?
Yeah.
Humor me. Yeah, good job. Oh, my kids are better artists than I am. All right, so there's a couple other things that are important. All right, they have cardiac muscle has the other important parts, components of regular skeletal muscle. Right? They are still going to do contraction using calcium, releasing calcium from the sarcomere. We still have to electrically stimulate the the sarcolemma, right? Sarcoplasmic reticulum. There's going to be myosin fibers. There's going to be shortening of the sarcomere in general. Okay, so that stuff is still the same, but there's some differences within it. All right, so there's your desmosomes and gap junctions that make up your intercalated discs, right? And you have your folded sarcolemma that is part of that, right? That's why I drew it in such a way. Okay, there are mitochondria to produce ATP. There are T tubules. All of those are similar or the same in terms of structure as skeletal muscle, right? So they're going to structurally be and work like skeletal muscle, okay? So we still have an H zone. They're striated, right? So you see the difference in H zone and Z disc. It shows you where the sarcomere is, and the sarcomere is going to shorten upon contraction, right? So all of that stuff is the same physiologically as what we've seen in skeletal muscle. Okay? This is what it looks like under the microscope, right? Those short perpendicular lines are your intercalated discs, perpendicular to the longitudinal axis of the cell itself. You have nuclei. Usually the nuclei is 1 or 2 nuclei, right? Most cardiac muscle cells are uninucleated. Okay? Basic questions on cardiac muscle cells. These are involuntary, okay? And so that makes up the myometrium. So we talked about, really that's the bulk of the cellular material that you have in the heart. Okay? The other stuff is what we call a fibrous skeleton, okay, of dense irregular connective tissue, okay, which makes up the valves, dense irregular connective tissue, the coordinate tendinae, right? A lot of the stuff that the cardiac muscle is going to live on is dense irregular connective tissue. Okay, and this provides a framework for the muscles to attach to, the muscle cells to attach to and to pull on, all right, and interact with one another. Forms the rings for the valves itself. Okay. The other purpose of this is that it acts as an electrical insulator. Okay. So I just said that the cardiac muscle cells are connected via gap junctions, which allows them to talk to one another electrically by the movement of ions from one cell to the next. Okay? This actually separates one area of the heart from the other. Okay? And this helps prevent the ventricles from contracting at the same time as the atria. Okay? That is important in terms of the sequence of contraction and how the heart works as a pump most efficiently. Okay? The normal sequence, the most efficient sequence, is that the atria contract first to push the maximal volume of blood into the ventricles, and then slightly after that, the ventricles contract to push blood out of the ventricles and out of the heart. Okay? That's the most efficient sequence. There are problems. If that sequence is disrupted, the heart doesn't work as efficiently, and there can be problems systemically. Okay, so you see the valves, etc. I was going to put a picture in here, but there's a whole YouTube video. So what you can do, you can call up your local butcher and ask for a heart. How many of you ever, their parents or relatives, brothers, sisters, hunt deer? Okay? So you can take the deer and you can get a heart, right? A fresh heart. And you can take away all of the cellular material that's there, okay, through a process, right? And produce what's called a ghost heart. And you end up with the collagen framework, which looks like a heart, but it's white and almost transparent, okay? That process is actually being used to produce new hearts. Okay? Now, what you end up doing is you can use that ghost heart, the framework that's there, and seed stem cells on it, and have the stem cells repopulate the entire heart, and eventually have a new heart. That's kind of where research is going. It hasn't been perfected yet, but that's kind of what they're doing. Okay? Questions? Okay? So, with this, the structure of the muscle tissue, so you have the fibrous skeleton, which is what the muscle tissue will attach to. Okay? And all of the cardiac muscle cells are kind of interwoven with each other, so you have this unique framework of a muscle. And the way that the muscle is arranged, the muscle fibers are arranged, is in such a way as kind of it compresses the volume or the inside of the heart, right? So it kind of, it's like a boa constrictor, but that compresses the heart itself, wraps around and ejects whatever inside, and shoots it out. Okay, I'm going to use Joe's water bottle as an example. My hand is the cardiac muscle cells. Okay, inside of this is an open volume, which would be ventricles. So when the cardiac muscle contracts, what happens? Where's the blood go? Out, right? That's all. So it's basically squeezing from the outside, but it's also arranged in a certain way that it squeezes the volume of the heart, of what's inside the ventricles, out in a certain direction. Okay? So everything's going to start, contraction's going to start at the apex of the heart, okay, down here, and it's going to cause the ventricles to squeeze from the apex towards the base. Now, in lab, you dissected hearts. And you saw where the aorta and the pulmonary trunk were. We went over that anatomy on Wednesday. Where is the aorta and the pulmonary trunk? They are at the base, right? So you're saying, this is going to be the aorta right here. Okay, right here's the aorta. Here's the pulmonary trunk. I'll do it in blue. Here's the pulmonary trunk. So what's going to happen is the blood is going to be forced out of those two vessels because you're forcing it back up, right? And that's also why you have the larger valves of the atrioventricular valves, which will help prevent backflow into the atria. Okay? And then you have the atria, which are the atrial muscles, which are different in terms of shape because they are going to contract to push blood down into the ventricles. All right, so everything structurally is set up to have a most efficient pump that's available to you. All right, so all of this is set up to have the most efficient pump process. All right, the heart itself is the muscle, cardiac muscle, needs nourishment, needs nutrients, needs oxygen. Absolutely requires oxygen. Okay, cardiac muscle itself depends upon aerobic respiration to produce ATP. Okay, so aerobic means what? Taylor? With oxygen. Okay, so it requires oxygen. So if you stop oxygen from getting to the heart muscle, it doesn't have a very high capacity to sustain that very long. Okay, so the coronary circulation we're talking about right here is absolutely imperative in nourishing the heart. But if you break it off, what happens? What if I stopped it right here? So that all the way down here now. Oh, that does not. All down here doesn't get nourishment. What's going to happen to that part of the heart? What's going to happen to those cardiovascular cells or cardiac muscle cells? They're going to die. Okay, in a very short period of time. Okay. And this is one of the problems, this is kind of one of the problems in the design of the heart. There's not an alternative circulation that can occur that would ensure that fresh blood, oxygenated blood is always getting to the heart. Okay? The coronary artery over here feeds this whole side. This coronary artery feeds this whole side. It's separate, right? There's not connections between those blood supply. Okay? As we'll see in a couple, probably like in a week and a half, we're going to talk about special circulation in the body. There's a things called anastomosis. Right? All right, or something like that. Okay. Anyways, what it does, it looks like something like this. It allows for blood to kind of go a different route, but still get to the same place, right? So if you were going to drive to New Haven, you could go down Route 10, right? Or if Route 10 was blocked, you could find an alternate route, right? You could go a back road. You could go out to 91 and go down that way, whatever it might be, okay? There's no alternate route for the coronary circulation. So if it gets blocked, that's why we have problems, okay? That's why we have heart attacks, okay? I'm not going to go through the different arteries. That's the same stuff you did. It's already in your book. It's already in your notes, so I'm not going to cover it, right? And then so we have coronary arteries, and they break off into different parts, left and right, and then which have other branches. And then we also have cardiac veins, which return blood back to the heart, okay? Blood that is deoxygenated, and it goes from the heart through the cardiac veins into what? Coronary sinus before it empties into the right atrium, okay? And the coronary sinus in this picture is shaded because it's on the posterior part of the heart, okay? Questions on the cardiac circulation? And it's, oh, no, I didn't want to do that. Okay? So, what do you end up with problems? Okay? We end up with differences and things that happen to these coronary. So, the idea of saying, don't have a coronary means don't, have a heart attack. Okay? These can lead to different problems, right? Because of the circulation, if it's blocked or the blood vessels are thick, can lead to problems, right? So we have atherosclerosis, the plaques narrow coronary arteries. So atherosclerosis is, so you have a blood vessel, looks like this, and then you end up with plaques that we'll make that a little bit smaller, okay? Plaques that form fat around it, leaving a smaller opening for blood to flow through. Okay? You have coronary spasms, a sudden narrowing of blood vessels. So these blood vessels have smooth muscle, so they'll narrow. Any type of narrowing, what does it do to flow? It decreases flow, right? So anytime I go from a vessel that is, this dilated to smaller, what happens to flow through that vessel? It gets constricted and can cause problems, okay? And both of those can lead to things called angina or myocardial infarction. Angina pectoris, it's just pain, it's usually on the left side of the chest, left arm or jaw, okay? And it's usually referred pain. What does referred pain mean?
Like coming from another place.
All right, so it's not necessarily, oh, my arm hurts. No, it's not your arm, it's not your bicep. It's kind of, the pain is being caused in the heart, right? So it's being referred to someplace else, all right? And treatments are used to induce a vascular dilation, right? Angina is not as serious as the second part, okay? But it is a prelude to having heart attacks or myocardial infarction, okay? Myocardial infarction is sudden and complete occlusion of coronary artery, okay? The myocardium is deprived of oxygen, And because of the nature of cardiac muscle to depend upon oxygen for ATP, it causes cardiac muscle cells to die. Okay? So you have pain in the chest pain, upper pectoral down to the left arm, and weakness, short of breath, nausea, anxiety, and sweating. And this says sudden and complete occlusion. This is I want to say that there are different, if you were a cardiologist, there are different levels of myocardial infarction, different levels of heart attacks, some that are prolonged because of a reduction in the flow of coronary artery where you're actually causing spasms, and there's a whole different, but we are just going to lump them all together for right now. Okay? Questions on that? Myocardial infarction, heart attack. All right, so we've set up the structure of the heart in terms of muscle. It depends, the function, the physiology of the heart depends upon the activity of that muscle, okay? And the structure of the muscle, in order for it to eject blood out of the heart in the right direction and most efficiently, okay? How does the heart know when? I mentioned this earlier about the sequence of contraction, okay? How does the heart control the sequence of contraction, okay? That's what we're going to cover next. And it has to do with the stimulation of how the heart muscle is stimulated and anatomical parts that help that stimulation, okay? So the sequence of contraction is actually preceded by a sequence of stimulation. These are muscle cells, right? So they depend upon electrical impulses to contract. All right? So what you see here are the the anatomical parts that help control the electrical stimulation of the heart. We're going to start at the top left, as you're looking at it, over the right atrium. That is the sinoatrial node. Okay? It's a node, specialized cells that help to determine how the sequence of stimulation occurs. Okay? So you have the sinoatrial node, which is the pacemaker. And so sinoatrial, so it means atrium. Okay? Sino refers to sinus. If you have a normal sinus rhythm, what do you have for your heart? How many of you watch Grey's ER, The Resident, any type of medical show on? That's like you've all seen and heard of a sinus rhythm. Blip, 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 beep. Yeah, right? You've all seen. Okay, right? We don't want the beep. Okay, so a normal sinus rhythm. That's the normal. So you're going to be sitting there, and every second your heart's beating. Okay, the normal sinus rhythm that is controlled by the sinoatrial node. Okay. The atrioventricular node is where? Atrioventricular. Right? It's right over the boundary between the atrium and the ventricle. Okay? You have atrioventricular valves, atrioventricular node. Okay? And then from there, you have a network of fibers, neuronal fibers, that spread through the interventricular septum down to the apex of the heart. Those are called Purkinje fibers. Okay? There's one particular part within the septum which is called the left and right bundles, also called the bundle of His. Okay? But it spreads down through the septum to the apex of the heart. Okay? Those are important. Why are they important? Coronary arteries, make sure I'm covering everything. Okay? The purpose of this is the heart's conduction system. This is going to control stimulation of the muscles by conducting the electrical impulses from one area to the next. Okay? So everything is going to start at the sinoatrial node every second. Okay? Because your heart beats at rest about every second, right? So every second, we're going to start over a contraction. And this sinal atrial node's going to fire. So that's number one. Okay? And I'm going to draw this out, and we're going to go through it again in a little bit. From there, that electrical impulse starts to pervade, and it spreads through the cardiac muscle cells over to the left atrium and down to the right atrium until it hits the atrioventricular node. Okay? So when the sinoatrial node, the SA node fires, the atria start to contract. Okay? Now that electric impulse reaches the atrioventricular node, and these nodal cells are lazy. Okay? They don't like to get up in the morning. They don't like to fire right away. So there's a delay that happens. Okay? There's a slight delay. Because why is there a delay? Why is it important that there's a delay? So the ventricles can fill with blood. Excellent. Right? So we just pushed, we contracted the atria. They push blood into the ventricles completely, right? So we want them to fill with blood before the ventricles contract, right? So that the most amount of blood can be pushed out of the heart at once. So there's a delay. And then it starts to contract from the AV node down to the bundle of His. Now, what did you notice about the separation between the SA node and the AV node? How would you describe that association? Is it direct or indirect? Indirect. Why? How does the electrical impulse get from the SA node to the AV node?
It doesn't touch them.
It doesn't touch them. It actually spreads through the muscle cells. Okay? Then it gets to the AV node, and now what do you see from there? Now the AV node starts to spread and goes to the bundle of His. What is that connection? Direct or indirect? It's a direct connection. Let me ask you, if you had the choice, would you prefer a wireless internet connection or a wired internet connection? For speed purposes. I want to move around. I like my phone. No. All right. For speed purposes. Wired. Okay. The connection between the bundle from the AV node to the bundle of His is a wired connection. Okay. So now through these Purkinje fibers, the electrical impulse spreads down through these fibers and around the ventricles in a very rapid transmission, causing the muscle cells in the ventricles to contract. Now what? What does that do? Contraction starts down here, spreads out towards, now what happens? Blood is pumped out. Okay? So we have the SA node fires first, delayed to the AV node fast transmission through the bundle of His and the Purkinje fibers within the interventricular septum down to the apex of the heart where contraction starts in the ventricles. Okay? Once that goes through, now what? We have to get ready for the next heartbeat, right? So everything relaxes for a fraction of a second, and then the sinoatrial node fires again. Okay? So your sinoatrial node is your pacemaker. Okay? Your AV node, we know where it is, extends where it extends, and then it goes from there. Okay? Excuse me. What controls this? So you have your normal sinoatrial node. Your SA node is your autorhythmic cells. Those cells are going to help control the fact that you have a heartbeat every second. Okay? If it were up to them alone, if it were up to the nodal cells alone, they would fire about 75 times per minute. That's a little bit higher than normal, right? Normal heart rate is about 60 beats per minute. Okay? So the actual, if you were to leave it up to the SA node by itself, you'd have a little bit of a higher heart rate than you necessarily do. Okay? So there are other things that help to influence heart rate. And basically it's your sympathetic and your parasympathetic nervous system. Do you guys need to like have a little peanut gallery outside? We good? Yeah. Okay. All right, so your parasympathetic and sympathetic nervous systems. One will help to increase heart rate. The other one will actually work to slow it down. Okay? And even the parasympathetic nervous system helps to control heart rate and keep it at a resting 60 beats per minute normally. So it actually works on the nodal system to hold it down. Okay? So in a lot of different responses to a lot of different things, you'll end up with control and what influences heart rate. Okay? So most of this comes from the cardiac center within the medulla oblongata. Okay? So the medulla helps control this, and it says information, all right, via nerves to the heart. Okay? I don't really care that you see it goes through the vagus nerve or anything like that. Right? But understand there are all of these inputs, stimulus inputs. So you have receptors in the aorta that say how much that will determine blood pressure. Like those are baroreceptors. Chemoreceptors for oxygen. Chemoreceptors for carbon dioxide. You have more pressure receptors within the right atrium. Okay? And they will help control how fast the heart beats and how strong the heart beats. Okay? All of this is based upon what we call cardiac output. Okay? Cardiac output. I'll write that up here. Cardiac output is a set determination for the body, right? Based upon your size, your athleticism, etc. Okay? There's an amount of blood that has to be pumped out of the heart every minute in order for adequate nutrients to be delivered to all your body cells. Okay? So cardiac output is equal to your heart rate multiplied by what we call stroke volume. How much blood is pumped out of the heart every beat. Okay? So every time it contracts, how much blood is ejected. Okay? And then the baroreceptors and the chemoreceptors, they help to influence cardiac output and help it keep it at a normal or homeostatic set point. Okay? By influencing how fast or how slow your heart rate is and how strong your heart contraction is. Okay? And we're going to come back to cardiac output towards the end of the chapter, but I did want to talk about it now. So you have a cardio-acceleratory center. What's that going to do? Speed up the heart. Cardio-inhibitory center, it's going to slow it down. All right, and your vagus nerve and the glossopharyngeal nerve also have inputs into this. Okay? So, the stimulation. There are basically two things that happen in a heartbeat. Okay? We have the conduction system, what we just mentioned, right? That's going to initiate and propagate the electrical impulse through the heart. And then on top of that, the cardiac muscle cells have to respond to that stimulus, fire action potentials, and then contract. Okay? Those two things are what's going to occur. Again, the sequence happens where first? At the atria, and then through the ventricles. Okay? So, first, the SA node initiation action potential. And this is basically what I drew out already, just in the pictures that were in your book. Okay? And you'll see the spread of the SA node electrical potentials through the atria. They go down and to the left. Right? Down towards the inferior part and spreads over to the left atrium. Okay? And it spreads. What's next is going to hit what?
Maybe not.
It's going to hit the AV node. All right? We'll talk about the cardiac muscle cells for a second. Okay? They are muscle cells. So they have a charged membrane. They have a membrane potential. Okay? They will react to electrical stimuli. So when you get a change in electrical impulse across the cardiac muscle cells, that initiates an action potential, and they start to contract, and sarcomeres shorten. So SA node fires, electrical impulse propagates, those atria then, the cardiac muscle cells within the atria contract to push blood into the ventricles. Okay? Let's talk about how the node works. Okay? These are like neuronal cells. They're kind of like a mix between the two. Right? They are neuronal derived. So they fire electrical impulses similar to neurons. All right? In order to fire those electrical impulses, they have to have a resting membrane potential. What do you remember about the resting membrane potential of neurons from last semester? What was it? What was the number? -70. And what was threshold? -55. What do you notice about the resting membrane potential of these node cells? Emma? It's higher. And what is it closer to? Threshold. So is it easier or harder for these node cells to fire? Easier. Okay? That's important. Okay? And then they have this group of different channels that are important. Okay? So you have slow voltage gated sodium channels, you have fast voltage gated calcium channels, and voltage gated potassium channels. Okay? What was the ion that was responsible for movement, or what ion moved during depolarization of a neuron? Sodium. Okay? That is not the case in the cardiac muscle cells. Okay? It's going to depend upon not cardiac muscle cells, the nodal cells. These nodal cells are going to depend upon the movement of calcium. Okay? So you'll notice calcium's outside, sodium's outside, potassium's higher inside. All right, so what's going to end up happening, you're going to have a nodal cell is going to be stimulated, going to start to open the sodium channels, which is then going to stimulate the opening of the calcium channels. Depolarization occurs. We get the transfer of the electrical stimulus, and then repolarization occurs by potassium moving out. Okay? Potassium moving out. Okay? So, let me ask you a question. If you or your pet had an IV, an intravenous dose of potassium chloride, okay, which a large dose, what would happen to your heart? So I'm going to add potassium chloride. And I say this because this is what's going to be happening in Texas. I don't think Texas. Someone's executing someone. Okay? It would stop your heart. Okay? So, death by lethal injection. Okay? This is the lethal injection they're using. Okay? So, potassium chloride increases the extracellular concentration of potassium. Now the node fires. Okay? What can't happen? What can't happen? It can't repolarize, right? So if repolarization depends upon potassium moving out, okay, potassium's going to move this way, okay? It's going to help repolarize the membrane, okay, so that it can fire again. But if you have so much, if you overload the system with potassium, it's not going to allow the node to refire, and it stops the heart. Okay? So, SA node has what's called autorhythmicity. What that means, spontaneous firing. It has an auto rhythm, right? And it reaches threshold, okay? About minus 40 is the threshold in this, right? So membrane potential changes starting with sodium, but then it depends upon calcium, okay? So you end up, like, this was like this. It should have 1 bullet point, 2 bullet points, 3rd bullet point. So you have reaching, you reach threshold from initiation, you depolarize through an action potential, and then repolarization, okay? Sodium channels open up so that potassium can flow out, but it doesn't flow out, right? If you have a potassium chloride injection, you have high potassium, that gradient, all right, that concentration gradient is disrupted, and no longer will it flow out, and it causes the heart to continue to seize and causes cardiac arrest. Okay? Questions on the node and how it works. All right? Depolarization and repolarization. So at rest, like I said before, we have what's called vagal tone. All right? You're sitting there, if I had you all take a pulse and heart rate, all right, how many of you have Fitbits on with heart rate on? No. What is it? A lovely 75. Am I stressing you out? Okay. Some people are higher than normal. Okay. At rest, normally it's about 60 to 65. Okay. And that is kind of your parasympathetic nervous system holding it down because your SA node by itself wants to be about every 0.8 seconds, okay, and which equates to about 75 beats per minute, okay? So this is what it looks like, different parts, right? Your resting membrane potential. Going to reach threshold about minus 40 for the movement of sodium, but depolarization depends upon what movement of what ion? Calcium, right? So it's not just the movement of sodium, right? Depolarization of the nodal cell depends upon calcium. So if you didn't have calcium, would that be a problem for your heart? Yes, it would, okay? And then repolarization, the movement of potassium ions. Okay? And then this is what we've talked about in terms of the differences between the nodal cells and neurons. Depends upon calcium, right? Nodal cells have a pacemaker potential, right? They don't have a stable resting membrane potential. It kind of fluctuates based upon those slow acting sodium channels. Okay, so let's go through the 4 parts. I think that was next, right? Okay, the 4 parts. The 4 steps of conduction. Sinoatrial node fires. It spreads across the atria, all right, causing the atria to contract. That's step 1. All right. Then it goes to the V node, and the action potential is delayed at the AV node. All right. Like I said, those nodal cells are a little lazy. Okay. And then it passes to the AV bundle, also known as the bundle of His, within the interventricular septum. That is a fast transmission through the interventricular septum down to the apex of the heart. Okay? And there's left and right, and they go through Purkinje fibers, which are specialized neuronal fibers within the interventricular septum, down to the apex, causing contraction of ventricles, and you get the ventricular myocardium, right? Spreads that electrical impulse across the cardiac muscle cells to cause contraction. Okay? That happens every second in your heart. Okay? Every second it starts over. Questions on this? And so there's an electrical impulse that is propagated from right atrium down through to the left ventricle. Right? Yes, Taylor.
Does the SV node fire before or after the contraction?
The SA node or the AV node?
Oh, AV.
The AV node fires right before, right? So you have the delay. So the SA node, delay, and then the AV node fires, and then it goes all the way in through to the ventricle. So it's right, AV node fires right before the ventricles contract. Okay? And then it repolarizes. So importance is kind of where this is. Right atria, right side, down towards the left side. So next week in lab, you're going to take electrocardiograms. And we're going to go over electrocardiograms on Monday. But how many of you have ever had an electrocardiogram done? Never had the leads on? Yes? No? All right. Where do they put them? They put one up here, one on the right side, one on the left side. They put one down here on the left side. Why do they think they put them there? You need at least those 3 leads. Why? Where's the electrical impulse traveling? From right to left. So they're actually tracing the very minute electrical differences that are being transmitted from the right to the left. All right, and they're picking that up, detecting that, and then putting it into what looks like something like this. Right? And this is what's called a PQRST wave. That's what your electrocardiogram is. Okay? I'll draw it a little bit bigger. Okay, so PQRST. The P wave, what's the first thing that happens every heart rate? What's the first thing that starts? SA node fires. What do you think happens between where the SA node fires and the next big thing? Casey. What's going to happen? Why is there a space between those?
Delay.
There's a delay, right? So you have your delay. And what's this QRS? What's this big one? This QRS. What do you think that is? What part of this whole thing is the largest electrical difference? The largest contraction that's going to occur?
The ventricle.
The ventricles fire. The ventricles, usually we're right, it is depolarization of the ventricles, because they're the biggest part of the heart, right? The atrial kind of small. This QRS is depolarization of the ventricles. And what's T out here? What do you think happens next? So atria contract, ventricles contract. Now what? Reset. This is repolarization. Okay? So there's your AV node right there. Okay, this is ventricle depolarization, and then you have repolarization that occurs over there. We'll go over that again next week, so it's kind of out there, but I just want to say this is what it refers to. It refers to the electrical conduction of impulses across the heart, starting at the right atria and continuing down through the left ventricle. All right? Questions on that? We'll pick up on that on Monday. Have a good weekend. We'll talk. Your first test is 2 weeks from today, right? 2 weeks from today, and it's on the blood and the heart. We'll finish the heart probably by middle end of next week.